My name is Nicole McNabb, and I am a PhD student in the Department of Environmental Toxicology at UC Davis. In this presentation about climate change, environmental justice, and COVID-19, Part 1 focused on environmental factors associated with chronic health conditions, and now, Part 2 will focus on vaccine response and climate change. The COVID-19 pandemic is an unprecedented global public health challenge that has resulted in staggering human loss around the world. Although COVID-19 is widespread, its impacts vary dramatically among subgroups of people. In many parts of the world, communities of color and disadvantaged communities are disproportionately affected by COVID-19. Exposure to environmental pollution represents an important piece of the puzzle underlying the increased incidence and severity of COVID-19 in these communities. In many countries, there are significant racial disparities in environmental exposures to pollution. Vaccines for COVID-19 may have reduced efficacy in populations with higher exposures to environmental pollution, which unfortunately are the populations at increased risk for more severe COVID-19 health-related outcomes. Further exacerbating the interaction between environmental justice and COVID-19 is climate change. Climate change is increasing environmental pollution in numerous ways, and these impacts of climate change are disproportionately affecting disadvantaged communities. In an ironic twist, climate change is likely also a significant factor in the transmission of the SARS-CoV-2 virus from wildlife to humans. Vaccines help prevent severe health outcomes from viral and bacterial infections by training the immune system to develop a rapid response to protect against these pathogens. Central to the protective role of vaccines is the generation of potent antibodies and memory immune cells that can be rapidly deployed and amplified if the target pathogen is encountered again. Vaccines activate B cells to produce antibodies that circulate in the bloodstream and can block a pathogen from infecting cells in the body. Vaccines also activate specialized T cells that target and kill other cells infected by a pathogen. While there are limited studies on the impact of environmental exposures on vaccine efficacy, given what is currently known about how the immune system can be influenced by environmental pollutants, the efficacy of COVID-19 vaccines may be limited. Environmental pollutants can suppress the ability of the immune system to generate a powerful antibody or cellular immune response by inhibiting the production of memory B and T cells. Air pollution is a widespread source of human exposure to environmental chemicals. Studies conducted in both animal and cell culture models have shown that exposure to air pollutants can suppress the immune response to pathogens. These air pollutants include ozone, which is a harmful pollutant at ground level and is the main ingredient in smog, and PM 2.5, or fine particulate matter that comes from sources such as wildfires, wood-burning stoves, car engines, and coal or natural gas-fired power plants. Studies have shown that exposures to environmental levels of some other pollutants are associated with reduced vaccine efficacy. These pollutants include perfluoroalkyl substances, or PFAS, a group of man-made chemicals from many sources, including nonstick cookware, stain resistance products, photography, firefighting foams, fast food packaging, pesticides, paints, and shampoo. Arsenic, which is naturally present at high levels in the groundwater of a number of countries and is highly toxic in its inorganic form, and polychlorinated biphenyls, or PCBs, man-made organic chemicals used in many industrial and commercial applications that were banned in 1979 but are persistent and still found all over the world. This figure summarizes the potential for environmental exposures to influence COVID-19 vaccine response. The combination of blood antibodies and cytotoxic T cells is essential for a robust protective response against a pathogen. COVID-19 vaccines activate B cells to produce antibodies that prevent the SARS-CoV-2 virus from binding to and infecting cells. COVID-19 vaccines also activate a specialized T cell that can recognize and kill cells infected with virus. The process of activating B and T cells can be altered by environmental toxicants that are influenced by climate change, including PFAS, PCBs, arsenic, ozone, and PM 2.5. Climate change is increasing environmental pollution in numerous ways. Melting polar ice caps release persistent organic pollutants that are sequestered in them. More frequent and larger wildfires generate more air pollutants. Acidification of the oceans increases the bioavailability of toxic metals to organisms. And finally, the expansion of the range of insects is driving increased use of pesticides around the world. Climate change and the associated extreme weather events such as heat waves, drought, flooding, and wildfires increases the likelihood of transmission of emerging infectious diseases such as COVID-19 from wildlife to humans. One of the main drivers of this increased disease transmission is a loss of biodiversity. Climate change has resulted in an extreme loss of biodiversity over the past few decades. Since the 1970s, it's estimated that populations of birds, mammals, amphibians, reptiles, and fish have declined by 30%. This increases disease transmission because of changing predator-prey balances, reduced competition, and die-off of species that are suboptimal disease hosts, resulting in an increased density of reservoir species. Habitat loss due to climate change, changing land usage, and spread of human populations into more rural and remote areas increases the likelihood of human and wildlife interactions, resulting in greater exposure of people to emerging pathogens. This increased exposure creates the opportunity for pathogens to move from wildlife populations to humans in a process called a spillover event. The number of spillover events are increasing and are responsible for the majority of major human disease outbreaks worldwide, including the current SARS-CoV-2 pandemic and previous outbreaks of SARS-CoV-1, MERS, Nipah virus, HIV/AIDS, and Ebola virus. Monitoring of emerging infectious disease hotspots where changes in land use patterns and biodiversity make human-wildlife interactions more common, as well as surveillance of endemic wildlife pathogens, is now a critical step in preventing future viral disease outbreaks. The year 2020 brought into sharp focus the inextricable connection between climate change, environmental justice, and COVID-19. At the center of this confluence is environmental pollution. More and more scientists are making the argument that this perfect storm demands early, aggressive action to mitigate dramatic human loss. As the eminent scientist Jane Goodall remarked in a discussion on conservation, climate change, and COVID-19, if we carry on with business as usual, we're going to destroy ourselves. In conclusion, exposure to environmental pollutants whose levels are increasing because of climate change can reduce the efficacy of COVID-19 and other vaccinations to prevent severe disease resulting from infection by pathogens. Addressing climate change is essential for not only mitigating severe COVID-related illness and other environmental justice issues, but also for limiting future viral pandemics. Are you interested in learning more about these topics? Be sure to check out Part 1 of this presentation if you haven't already. For more information, scan the QR code below to download the full ebook about climate change, environmental justice, and COVID-19. Thank you for listening.